Okay, guys, let's get started. You know, my favorite is when I drop my kids off at school. They're in kindergarten, and it's this new school, and it's really crazy. And they are always trying to make announcements, and it's just like there's this constant din of kids and teachers talking, even while the principal is trying to make announcements. And I always think to myself, how lucky I am that you guys just stop talking. I guess that happens sometime later in life. Kindergarten, first, second grade, they don't stop. Okay. Exam, I meant to bring them, but you'll get it. You can get them in recitation. You already know your grades. The average was a 79. Oh my God, I've never in my life seen that. Yeah, I mean, my goal is a 75. If I have a 75 average, then I did my job. 79 means you guys are rocking it, so good job. As you can see, that kind of is represented by the number of people in the 70 to 79 column. And the number of people who got over 100, so that's pretty awesome. Are you guys confused? You're just discussing? Everyone's going, I got a 99, I got it. Are you guys confused? Okay, is everyone excited? Yeah, that's awesome. Okay. I'm glad you're excited. Uh-oh. I'm going to make you do the Pledge of Allegiance in a minute. Sorry, too much time. Kindergarten breakfast. Okay. Exam 2 is changed to 10/28. There was discussion about whether or not everything would be pushed ahead. There's no way we'll be done with this by Monday. There's no way I'm giving another exam on Monday. So 10/28 is our goal. There's a small chance if we're lagging behind by early next week, I might make it on Wednesday, but the goal is 10/28. If you are in this below 60 range, which is not that many of you, but you exist, you must come see me. Because if this continues, you will fail. And we don't want that. No one wants that, especially people who are trying to graduate, right? Okay, so please email me and say I am one of those people. Actually, those people are probably all not here, right? Just say I'm one of those people, I need to see you, I don't know what I did wrong. Okay, let's move on. So we started the other day the signaling stuff, and we played a game of telephone. That was fun. I think we should do that more often. So we started with a generic signaling pathway. Just, you've got a signaling molecule, a receptor, a bunch of signaling proteins, and then something that activates a transcription factor to go to the nucleus and activate gene expression. If you get the gist of that, you'll be fine. That's your big picture you want to focus on throughout this whole entire unit. We did a few example pathways that may have made you scared, but remember they were example pathways. And as we start talking about this, I want you to start to think back to those pathways, and we'll go over other ones in the future, but start to realize which part of which pathway is which thing that we're talking about. So, for example, we talked about different types of extracellular signaling molecules. So you have to think, okay, what's the purpose of that, right? We talked about these secreted hydrophilic and secreted hydrophobic signaling molecules, and we also talked about how you can have just communication between two receptors on one cell, so there's not an actual molecule being secreted, it's just two receptors binding to each other. That's going to activate a signaling pathway. We started talking about types of receptors, and this is how we're going to do it. We're going to start outside the cell and move our way down and just talk about the general ideas of the types of proteins that are involved in what they do. And I just want you guys to start organizing things based on categories. Like, okay, there are different kinds of signaling molecules. They tend to bind to these types of receptors. They tend to activate these kind of proteins that do these kinds of things. And then that activates gene expression. It could be as simple as that or as difficult as memorizing a million pathways. Do not memorize a million pathways, even though sometimes it might feel like I'm giving you a million pathways. Okay? So we started with extracellular signaling molecules and then types of receptors, and that's where we're going to pick up. Right? So there were 3 classes of cell surface receptors. Notice it's 3 classes of cell surface receptors because nuclear receptors are not located on the cell surface. And we went over nuclear receptors the other day also. So there are ion channel coupled receptors, which I'm not spending too much time on. G protein coupled receptors, which I'll call GPCRs sometimes. And enzyme coupled receptors. So this was the ion channel coupled receptors. These are mostly involved in the signaling, rapid synaptic signaling between nerve cells and target cells. So signaling is inducing a conformational change that affects the ion channels, either open or closed, and sometimes these are regulated by GPCRs. Then there are GPCRs. These are G protein coupled receptors. Automatically, you just say G protein, GTP binding, molecular switch, GTP bound on, GDP bound on. This should just come to you. Boom. Right? It's Monday, but it's Monday at 3:30. When it was Monday at 9 a.m., I was like, okay, guys, you get it. It's Monday, it's 9 a.m. It's 3:30. Did you all have your coffee? No. Actually, I can't drink coffee after about noon. Or it's bad news. Bad news. It's crazy. I just can't sleep, and then I'm crazy the next day. Okay. G protein coupled receptors, where you have the receptor that's going to bind your extracellular signaling molecule, and then you'll have an inactive G protein, and then there'll be some kind of enzyme or ion channel that's regulated by the G protein. So molecular switch turns things on, turns things off. So it can regulate ion channels or membrane-bound enzymes. And then there are the enzyme coupled receptors. And they have either intrinsic kinase activity or an associated enzyme activity, meaning they're binding to a kinase versus having a kinase domain. You asked me a question over the weekend about that, right? Yeah. So I got a question over the weekend about the quiz question about whether phosphorylation, what was it? Phosphorylation of the cytoplasmic domain of RTKs? Yep. And what's the outcome? And I was trying to get you guys to compare and contrast the difference between the receptor tyrosine kinases and kinase-associated receptors, which are 2 different things. One has the intrinsic kinase activity, and one's going to have a kinase bound to it. So when you're studying, I want you guys to start thinking about those differences and how they're regulated slightly differently. And hopefully that'll come through during these lectures. So here's it showing there's this kinase domain here, and signaling results in ligand binding results in activation of those domains by bringing the cytoplasmic tails close together. And then here it's showing how you have this enzyme that's associated somehow with the kinase, with that receptor, and the signaling molecule activates that. Either way, you have a kinase being activated. Okay, so I'm going to remind you just quickly about molecular switches because we're going to talk about GPCRs. And if you forgot, hopefully no one forgot about molecular switches because how many examples of molecular switches have we had so far? 2, right? RAN was one and RAB was one. So now we're going to have another, did we do another one? Oh no, RAS. RAS hasn't come up yet. There's going to be another R. So in this case, but just a reminder, this has to do with how you're going to have it either in an on state where you have GTP bound or an off state where it's GDP bound. And you could also think of ATP binding also as a molecular switch in a way, because sometimes phosphorylation will activate things. That's not all the time. Phosphorylation can also be inactivating, as we shall see when we do cell cycle. But it can also be activating. So you can think of that similarly. Okay, so that's molecular switches again. And so then I want to, again, I introduced this earlier, but just make sure everyone's understanding the concept of domains. So this is a nice figure that shows how domains are different parts of a protein that do different things. So for example, here is your receptor in green, and your signaling molecule binds to it, and it has this Y, which is a tyrosine, that's been phosphorylated, and it's binding to the SH2 domain of this other protein. And SH2 domains are just a part of a protein that binds to phosphorylated tyrosines. That's the job of the SH2 domain. Okay? Then there's a kinase. So this protein, this green protein, what is that? It's like an aquamarine. Green, blue, I don't know. Signaling protein 1 here. So this is one protein, but has 1, 2, 3, 4 domains. It has this pH domain, plextrin homology domain, which binds to phosphorylated inositol phospholipids, and you see it's bound there. And then you have this PTB domain that binds to phosphotyrosine, but in a different situation. And then there's SH2 domain that binds to phosphotyrosine. Then there's a kinase domain, meaning it has what kind of activity? It can phosphorylate things, right? Kinase domain has the ability to be a kinase. It can phosphorylate things. That's one protein with all those business areas, right? And so you could see how phosphorylation of things regulates all this stuff happening, right? Because if you have signaling and then you end up with phosphorylation of your cytoplasmic tail at this tyrosine, it creates a docking site for signaling protein 1 because it has this SH2 domain. It wouldn't bind if it wasn't phosphorylated, right? Are you guys bored out of your minds? Sometimes I feel like I'm just talking. I can't stand it when I just keep talking for like an hour and 15 minutes. Okay. Who loves SH2 domains? Okay, you better. Love it. Okay. So then there's another signaling protein that also has these domains. They all regulate. It's all about regulation of binding of things. Yes?
So the whole protein, all those proteins add together, the SH2 domain, or
Right, so, okay. So see how that right there says SH2? That little square. Oh yeah, you need glasses. That's the problem. Or a telescope.
So phosphorylation can be used.
Opera glasses, sorry.
Can they be used? So phosphorylation here is used for the docking of proteins, but can it be used at the same time to block?
Right. So the question is, phosphorylation here is used for the docking of proteins. Can it also be used to block binding? Yes. But you would not have an SH2 domain, because SH2 domains, by definition, like to bind phosphotyrosine. So the way the protein has folded creates the perfect binding pocket for phosphorylated tyrosines. In other situations, you might have like the NLS example, where you phosphorylate your NLS, you then can't find it. So that's a very good question, because people always think phosphorylation activation. But no, it's so much greater than that. And that's why I really try to emphasize it's changing the shape. It might change the shape of the protein so that it can bind something, and it might change the shape of a protein so it can't. And that's why on an exam I'll always say something like, it's a deleterious mutation, so so you at least know which direction to be thinking in. What else? So this is kind of how signaling starts. You'll have your signaling molecule bind to your receptor, and then this phosphorylation will happen, which will recruit proteins, which will then recruit other proteins, and then there'll be a kinase domain that'll somehow become activated, and they'll phosphorylate other things, which will create more docking sites and recruit other proteins. And then with the whole point is to relay the signal all the way to the point of sending a transcription factor to the nucleus to activate gene expression, pretty much, or to affect gene expression, I should say. Okay, so does anyone have questions about the concept of domains? Because every year people really are lost on it, and every year I tweak this part of my lecture, hoping to get you from another angle. Does everyone get it? It's different parts of a protein that do different things. Okay. I'm seeing some people nod. That's good. Nodding is good. Okay. So here's a practice problem for you. So you're studying a protein that's expressed by a newly identified virus that infects frogs. I have to give equal opportunity to the people who like animals other than humans. It's a virus that affects frogs. You have the protein's amino acid sequence, and you run a BLAST search to learn more about the protein's identity. You guys all know what BLAST is, right? Okay. No? Who said no? Thank you. Yay! BLAST is when you put a protein sequence into a database, and it compares against all the other, it's into a more like a search engine kind of thing, and it compares the sequence against all the other sequences in the protein databases out there and finds similarity. And so you do this to learn more about the protein's identity. And the search returns a number of proteins, and they're usually ranked in order of identity, so how similar the protein is to other ones in the database. So you've got this unknown protein, you blasted it. It brought back all these other proteins, including one with 98% identity that has kinase activity and an SH2 domain. So you know nothing about your frog protein. But blasting it showed you that it has these domains that are quite similar to what's in another protein. That's an SH2 domain and a kinase domain. What hypothesis can you form about your unknown protein, and how can you experimentally evaluate your hypothesis? So take a minute. You're alone. What happened? Wait. Okay. Sorry, I got confused. So you guys take a moment to discuss. Do you need clarification on the question? You feel okay? I'm going to leave it up here. Discuss. I'll circulate. I want you to start thinking about domains. Okay, guys. I'm going to start doing equal opportunity. I always go up around one way and then I get stuck over on this side. Start going around. Okay. So the question was referring, I'm sorry my pointer is bad. Can you see it now? No. What if I just use my finger? The back row doesn't like my pointer, which I totally get. It's a bad pointer. I'm sorry. How about the front row? Do you like my pointer? With middle rows. When do you stop seeing my pointer? Who sees that?
I can see it all.
Yeah. Okay.
When you move it, you can't see.
I'm just gonna have to get a big flashlight. Okay. So what I was trying to get you to think about was what can you infer? So you found a protein that's similar, and it has an SH2 domain, and it has kinase activity. And so you need to say to yourself, what did Dr. Ehrlich say an SH2 domain does? What is kinase activity? What kind of protein might have an SH2 domain and kinase activity? What cellular process might this protein be involved in? And then from all that, you would say, well, my frog virus protein, it probably binds to this. It probably does that. And you are on the wrong side of the classroom. Yeah, okay, just making sure. Sorry to get thrown out.
Sorry.
Then you have to design an experiment. So what does kinase activity tell you about the function of your protein? Huh? So I'm hearing enzyme coupled receptor, maybe. What else? So phosphorylation might be occurring. Okay, so I got enzyme coupled receptor from down here because enzyme coupled receptors are often have kinase, not coupled, but enzyme linked receptors do have kinase domain. And then yes, so some sort you would say, okay, does this gene have kinase activity? So that would be one thing you could evaluate. SH2 domain, who picked up on what that does? Huh? Binds to the cytosol's tail. In this picture, yes, but based on what? What specifically does the SH2 domain bind to in the cytosol's tail? Yes. Phosphorylated tyrosine. So you would say, oh, I wonder if this protein can phosphorylate things, and I wonder if it binds to phosphorylated tyrosines. And then you might say to yourself, well, proteins with SH2 domains that have kinase activity often are involved in signaling. Often they bind to cytoplasmic tails. And then you can start to think about what your viral protein does, because you don't know anything about it. So it could be anything from a cytoskeletal protein to a histone. I don't know. There's so many types of proteins out there with different functions. So these little bits of information can help. So how would you experimentally evaluate your hypothesis? So we just formed hypotheses, right? Did you catch that? Yes. Who didn't catch any hypothesis forming? Yeah. So the hypothesis was maybe it has kinase activity. So maybe it can phosphorylate things. And it has SH2 domain. Maybe it binds to phosphotyrosine. That's our hypothesis. We don't know. No one did the experiment. Now you need to design an experiment to test these things. So what kind of experiments might you think up that could evaluate whether something phosphorylates something? Yeah? No. You don't need to know how to do science to kind of make something up. Yes?
Alter the binding site, so what typically is supposed to bind together binds to something else, and then you see the process involves through.
Alter the binding site. So this is the kinase, or is this SH2? Huh?
Whatever would inhibit whatever's going on.
Alter the binding. So are you saying mutate something?
Yes.
Okay. Mutate something. We're going to stick with kinase now. Mutate your putative, so your hypothesized kinase domain. Mutate it. Yes.
This is a bad question. I told my last one. Do kinases carry around phosphates with them? Could you tag a phosphate so that you can see where it goes?
Okay, okay. Kinases do need to bind phosphates to do what they need to do. Okay, so let's merge your two ideas. Mutate your kinase domain. See whether it can transfer radioactive phosphate to some known substrate. That's a tyrosine. Yeah? Yeah? If you mutate it should lose the activity. And it should have activity when it's not mutated. Okay, SH2 domain. Who wants to come up with something like that? Let's see. I'm getting all the same 1, 2. I'm going to pull out my list and find out who's not here. Nina. You changed your hair. I can't tell you. So what kind? So you think there's an SH2 domain? You know SH2 domains bind phosphotyrosine. What kind of experiment can you design to evaluate whether your hypothesis is correct? So so somehow make a phosphotyrosine and see if the SH2 will bind to it. Okay, can do that. Yes?
Could you read the SH2 domain and see that would affect the
Cut off or mutate your SH2 domain, see if it could still bind to a phosphotyrosine. Yep. Do you want to add?
I was just saying, couldn't you just like mutate the SH2, see if it binds to
Yeah, mutate it, see if it binds to phosphotyrosine. Yeah, so you're all right. This is how you do science. When in doubt, if you think something does something, get rid of it or mess it up and see if it still does it. Okay, let's move on. That was fun. Okay, so through it all, don't lose sight of the big picture. So I put up my big picture slide here for a minute, and just remember that this is what's happening. Extracellular signaling molecule receptor, intracellular signaling proteins relaying message to some kind of something. And I mostly focus on the gene regulatory protein and activating gene expression, but it also will activate, it will also affect cytoskeletal proteins such as actin and help with cell movement, which we will learn about very soon. And it will also affect metabolic enzymes, which I don't really talk about at all in this class. So don't limit yourself to gene expression, but that's a very easy thing to visualize. Okay. Okay, so let's start with GPCRs. Who wants to start with GPCRs? Should we do that? Sure. Okay, G protein coupled receptors. So they're chemically and functionally diverse, but they all have a similar structure. They're these huge 7 transmembrane pass proteins. Where are they translated? Yay, in the ER. Very good. And they all use G proteins to relay their signal. So they're GTP regulated. So this class has been going, it's like the Nobel Prize Committee and I are kind of all on the same page. Last year, at the same exact exam you guys just had, no. It was, I guess the timing was different, but it was like the same day I was teaching GPCRs, the Nobel Prize was awarded for GPCRs. And then this year, it was on the day of the exam about vesicular transport that the Nobel Prize was awarded for vesicular transport. I think about 2 people got that. So the Nobel Prize website has really nice figures to explain what the Nobel Prize is for, so I highly recommend playing around on there. So this is straight from them. And so it's explaining the activity of GPCRs. So you've got this 7 pass transmembrane receptor. and it's going to bind to whatever signaling molecule, in this case a hormone. And then the receptor, the binding of the signaling molecule is going to alter the shape of the receptor, and that's going to recruit the G protein and activate it. And then the activated G protein, the alpha subunit, is activated and goes down the signaling pathway in some way. So the receptor it's a G protein coupled receptor. So the G protein is not actually part of the receptor, it's coupled to the receptor. So when it's activated, the G protein becomes activated and goes on to activate something else. Okay? So they're GTP binding proteins, which you should think it binds GDP. It has GTPase activity. It's inactive when it's GDP bound. It's active when it's GTP bound. And the conformational change allows interaction with the target protein. So usually you have a receptor, and then the G protein gets activated, and once it's activated, it goes and activates something else. So it's a trimer, so it's made up of 3 different subunits. And so normally when it's inactive, it's just hanging out there separate from the receptor, and whatever other protein it's supposed to be activating, it'll also be close by, but it will not be active. And once the signaling happens, you get a conformational change, and then you get activation of the G protein, and then that G protein goes and activates this other either ion channel or plasma membrane associated enzyme. Okay? So when the GPCR is activated, so this should all just kind of make sense because we've learned a lot about this. When the GPCR is activated, you're going to get release of GDP and then GTP combined, right? That's kind of usually what happens with G protein activation. And then the signaling is active until GTP is hydrolyzed, because when GTP turns it on, GDP is off, right? So in this case, GTP hydrolysis is stimulated by the GTPase activity of the G protein alpha subunit. And it can be regulated by binding to the target protein, so whatever it's activating, or by some other regulator, which very creatively is called regulator of G protein signaling, or GS. Isn't that creative? Not like the Drosophila folks that name things frizzled and disheveled and wingless, and we just regulator of G protein signaling. So you can think about it, you've got this inactive conformation when it's GDP bound, and then addition of extracellular signaling molecule allows the replacement of the GTP for the G protein, and then it's turned on. So you can think of it as one protein's acting as a GAF and another's acting as a GAF, right? What's acting as a GAF? GAF is usually what activates GTP hydrolysis, right? So that would probably be the alpha subunit. And then the GAF is the signaling molecule, right? Because it's really the signaling molecule that activates the G protein, and then you get release of your GDP, allowing GTP to bind. Is this similar to RAN-GAP and RAN-GAF a little bit? Really about kicking off GDP and allowing GTP to bind, or hydrolyzing. Same function, right? GAPs and GAPs are always going to do the same thing. They're just specific for whatever G protein they're regulating. Okay? Okay. But what is the GPCR doing? That's what you're asking, right? So some G proteins regulate cyclic AMP production. Cyclic AMP is a small intracellular mediator, so it's this molecule. It's basically going to activate other things. that activate signaling. And an extracellular signal can cause an increase of cyclic AMP, and then cyclic AMP will stimulate some kind of downstream response. And I'll give you an example. Well, here's a few examples from the textbook. Adrenaline, this one, glucagon, thyroid stimulating hormone, they all activate adenyl cyclase to make cyclic AMP, stimulating triglyceride breakdown in fat cells. So that's one example. Just something to acknowledge is that there are different kinds of G proteins. Some can turn things off, some turn things on. They're stimulating, and some turn things off. They'll inhibit adenyl cyclase by regulating ion channels. So then what is, so you get this, so hormone or something is binding to a receptor, activates a G protein, and that activates some other thing which would be adenyl cyclase to increase cyclic AMP. You got the cells full of these small mediators, and what are those doing? They're binding to some enzyme and activating. In this case, you have pKa, which is normally inactive because it's bound to some regulatory proteins. And when cyclic AMP floods the cell, it's going to activate the catalytic subunit. Actually, it's going to bind and actually release the catalytic subunit. So this is how the whole thing would look together. So you're going to have your signaling. So you'll have your signaling molecule binding to your GPCR, and then you're going to have your G protein activated, which will activate your adenyl cyclase, and then you'll have cyclic AMP production, which will activate your pKa, your kinase. And then that activated pKa goes into the nucleus, and it can, what's pKa going to do? Just guess by its name. It's a kinase. It's going to phosphorylate something. So it actually phosphorylates a transcription factor, and that allows it to activate gene expression. That whole thing, all of that stuff was really all the same, where you just have signaling molecule bind, activate gene expression somehow. But the only difference is this is through G protein that's activating this membrane-bound adenyl cyclase. Okay? So you get this increase in cyclic AMP, activation of a kinase, goes to nucleus to activate gene expression. Any questions about those?
So the cyclic AMP is actually, that's really the mechanism for it turning. It's not really the gaps. There's like two mechanisms that have to go off with that.
So the comment that I just got is there's more than one mechanism of regulation here. It's because it's a cyclic AMP that's regulating the pKa, but the cyclic AMP won't be turned on unless the GPCR is activated, or the G protein is activated. So you can think about things as like every step of the way is this on or off. And every signaling pathway, it's not just signaling molecule binds to receptor, boom, gene expression. I mean, I make it out to be that easy, but every step of the way, you'll have something that needs to be phosphorylated or activated or something. And in this case, you have to make cyclic AMP to activate pKa, and then pKa has to be active so it can activate Krebs in this case and activate gene expression through that way. So in any of these cases, something can go wrong. So if something's wrong with your pKa, for whatever reason, it can't be activated, you still won't get gene expression despite binding of a signaling molecule to your receptor. I'm not convinced. Yes? Do all GPCRs follow this pathway? This is one example. But you could say, many GPCRs signal through these small intracellular mediators, such as cyclic AMP, but also calcium is another example. So you could say that.
This is just a general example.
This is one very general, simple example. Yeah, good question. Anyone? Any other questions? Any thoughts? Anyone want to tell me about their favorite GPCR? You know, so I'm so thankful that I teach this because my sister just got her PhD in GPCRs. So she would call me and she would be like, I'm having this trouble with my experiments. Thank God I teach this because I wouldn't know anything about this if I could teach it because I don't work on GPCRs. So I know it like one step above what you know it. Only one step. She works on prostaglandin receptors, neuroscience. Okay, any questions? Or I shall move on. Good. So you might ask, why use these small intracellular mediators, and you can think of it as they're very fast. They're very fast response because really usually you're in calcium you're just opening a channel, or with cyclic AMP you're activating an enzyme that's going to quickly generate a lot of cyclic AMP, and so you can have a very fast response. So this is just showing a cell that's responsive to serotonin and at time 0. So what they're showing is cyclic AMP in this. And so blue is low and red is high. And so they add serotonin and within 20 seconds you've got increased amounts of cyclic AMP. And mind you, that's like step 3, right? Because it's like serotonin binds to receptor, G protein activated, G protein activates adenyl cyclase, flood cell with cyclic AMP, and then you can guess that's going to go on and activate probably PKA and some kind of gene expression. Yes?
So like muscle contraction, I'm guessing like with the calcium, I guess calcium is playing the cyclic AMP role.
Well, okay, so I teach this, so that's different. Muscle contraction. Who's are you taking advanced phys or AMP or I don't know? So calcium. Calcium?
Yeah,
I'm thinking calmodulin, tropomyosin. Come on, guys. And then you're moving the thing. Talk to me in a few weeks when I teach it again. It's different because that's not signaling, that's activation of motor proteins. So it's different, but I like that you're trying to bridge them. Okay, any other questions? Okay, so just for fun, G proteins are targets for bacterial toxins, specifically color toxin and pertussis toxins, and they target different types of G proteins. One targets stimulating G protein, and one targets an inhibitory G protein. And so these toxins are actually used in the lab for research if you're trying to characterize a GPCR. So color toxin, it actually ADP ribosylates the alpha subunit of a stimulating G protein. And so you'll have GTP, it will be on and it activates adenyl cyclase, and the color toxin will keep it on. And so that since it's kept on, you'll have this elevated cyclic AMP levels in the intestinal epithelial cells, right? Because that's what's affected by color. And that results in efflux of chloride and water into the gut, and that's diarrhea. That's how you get diarrhea. And then pertussis toxin also catalyzes the ADP ribosylation, but it's of an inhibitory G protein. So it keeps the G protein off. So they're both useful. Lab tools for if you know there is a response that's dependent upon a G protein or a GPCR, you can study by using a specific inhibitor whether it's an inhibitory or a stimulating G protein. Okay, other roles of this is kind of gonna answer the question in the back. Other roles of GPCR. So cyclic AMP is just one example. Others, activation of plasma membrane-bound phospholipase C. So that's an enzyme, a plasma membrane-bound enzyme that regulates processes such as glycogen breakdown, amylase secretion, muscle contraction. Oh, muscle contraction there, but different through IP3 or PKC-induced effects on intracellular calcium levels. So I feel like now we got to go back to your question later. This is going to turn into a stump the teacher question. Let me think for a minute. I'm about to give you a stump the teacher question if I can formulate it right. Everyone, turn on your listening ears. So earlier Tyler asked about calcium levels in muscle contraction and whether it was through GPCRs, right? And I said, well, it's different, blah, 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 calcium, calmodulin, motor proteins, blah. So I didn't connect the two, but then here I'm telling you that GPCRs do affect muscle contraction by affecting intracellular calcium levels, which is directly contradicting what I was saying. So, your job is to tell me how GPCRs are affecting muscle contractions and affecting calcium levels, because I'm thinking like calmodulin, tropomyosin, and all this about motor protein function, and it's not, I'm missing something somewhere. My students are always teaching me. So I want you guys to tell me how muscle contraction is regulated by GPCRs and how that affects calcium levels. Do not tell me how muscles contract. Do not tell me anything but how GPCRs and calcium levels and all that together play a role in muscle contraction. Okay? And so that means you have until 5-ish tomorrow to email that to me. I don't repeat anything. So this is in response to Tyler's question about calcium levels and how increased calcium levels involved in muscle contraction. Is it through GPCRs? And here it says, I guess, yes, it is. Yes, but. Oh, God. I don't know. Okay, the skeletal muscle only from the physiology experts in the back. GPCRs, calcium, skeletal muscle contraction. How does it work? If I have to repeat it again, I'm going to forget. Okay, and don't look at, and that's the other thing. Do not ever email me the answer while you are in class, because people like to do that sometimes, and I don't like that because that means that you were not listening to me, and you are Googling the extra credit instead, and that 1 point you gain from your extra credit will not help you if you fail this exam because you mismade your concept. So always wait until at least 5. Just so I can pretend like you're hanging on every word. Okay. Okay, so other roles of GPCR is regulation of ion channels in the plasma membrane, directly or indirectly. And that's that. Okay, so here this is the release of calcium. Oh, I totally know the answer to my own question. I'm skipping this so you can look it up yourself. Either that or I take away the extra credit. Yeah, okay. Ah! Okay, guys, I'm not on my game today. Yes?
If somebody asks a question, you all get an answer. Can we all get
You got emails me. Because I love getting emails from my students. Okay. Okay, guys, we're getting out of control. Good thing I'm not being observed today. Okay, no one. Woo. Okay. Smell and vision are also regulated by GPCRs that regulate cyclic nucleotide gated ion channels. And so this is just using, so with the cyclic nucleotide, it's literally a nucleotide, but you'll have cyclic AMP, cyclic GMP. There are different of these small cyclic nucleotides that regulate things. So the olfactory receptors are GPCRs, and they bind odorants and act through cyclic AMP. An increase in cyclic AMP opens cyclic AMP gated cation channels, and that allows an influx of sodium, depolarization of olfactory receptor neurons, sending a nerve impulse to the brain. Oh, I guess that is it for GPCRs, really, already. Okay, so, this is so, no, okay, sorry, guys. So let's just talk a minute about receptor desensitization, because you do not want to signal forever, right? Yes?
Just like when you walk into a room, it has smaller candles for 15 minutes and less smaller.
Oh, like if you get a smell in your nose and it sticks with you forever?
No, like it goes away, you don't smell it anymore. Is that the desensitization?
Oh my God. They're saying yeah. I don't know, have I ever had that happen to me before?
That's why your house doesn't smell when you go in.
I always smell my house when I'm okay. I think things are getting out of hand. Too relaxed. So interesting. You guys are always making me think about things in a way that I've never thought about. Okay. Oh, I'm so glad that you're here for me. So some of this receptor desensitization we've learned about before, like this one. Oh wait, no, for those in the back who don't like my pointer, this one, right, where you have your signaling molecule and then you get endocytosis, or you can get receptor sequestration or downregulation, but you can also get receptor inactivation, and then you can get inactivation of your signaling protein. So here would be your GPCR, and so what would that be? Your G protein, right? Something there could be inactivated. And then you could also get production. So you can also get your signaling actually results in production of an inhibitory protein. So GPCR desensitization depends on phosphorylation and binding of the receptor by arrestin. So you'll have an activated receptor, and then it'll get So it'll get phosphorylated, and then basically it'll create docking sites for recruitment of arrestin, and then it'll be unable to. I'm sorry, guys. So this is your receptor, and so it'll be unable to interact with your G protein because you'll phosphorylate your receptor. And remember, once you have signaling through your receptor, Then the G protein gets activated, interacts with your receptor, and then activates the other protein. And so now we'll be unable to activate your G protein. Okay, I think I'm going to stop here today. We'll continue Wednesday. Don't forget the stuff this speaker that I gave away.
Professor.